Thank you. So now we have our last speaker of the session. André Miranda is a student from the joint MD-PhD program between the University of Minho in Portugal and Columbia University in New York. In the past 3 years, he conducted his graduate studies entitled Lysosomal Dysregulation in Neurodegenerative Disorders in Dr. Gilbert Di Paolo lab at the Top Institute for Research on Alzheimer's disease and the aging brain in Columbia University. He has returned to Portugal this academic year to complete his last year of medical education. Also, if you want to discuss further his work, is in poster 67, right, André? Okay.
So before all, I would like to thank the organizing committee for this great chance of presenting my work firsthand with such an amazing audience. So without further ado, today I'll talk to you about the candidate role of exosomes as biomarkers for neuronal lysosomal dysfunction. So we know that the endosomal and lysosomal system are tightly linked to neuronal fitness based on the fact that rare mutations in key endolysosomal regulators typically result in lysosome storage disorders that present with an aggressive childhood onset neurodegeneration. However, more recently, other milder partial loss of function mutations in additional endosomal regulators have been associated with late onset neurodegenerative disorders, namely Alzheimer's disease, Parkinson's disease, and frontotemporal dementia. And mutations can also occur in aggregate-prone proteins. And altogether, these significantly impair lysosomal lytic capacity, eventually leading to defective cellular clearance and neurodegeneration. In the case of Alzheimer's disease, mutations in 3 particular genes, namely amyloid precursor protein and presenilins 1 and 2, cause early onset forms of the disease. However, late onset cases account for over 90% of Alzheimer's disease cases, and recent genome-wide association studies have highlighted 3 clusters of genes as potentially important risk factors for the disease. And these can be sorted into endosomal-related genes, lipid metabolism genes, and immune response genes. In an attempt to characterize the role of lipid metabolism in Alzheimer's disease, the lab I was working in, Dr. Gilbert Di Paolo's lab, conducted a full lipid profile of the brain of AD patients and compared it not only to controls, but also transgenic mouse models of Alzheimer's disease. And at a first sight, we detected the accumulation of sphingolipids in Alzheimer's disease patients. And these lipids are typically associated with lysosomal storage disorders, namely Niemann-Pick type A, B, and C, and other sphingolipidosis. However, out of the all lipids analyzed, only one of them behaved the same way in AD patients and animal models of the disease. And this lipid was phosphatidylinositol 3-phosphate. This phospholipid is particularly enriched in the endosomal system. It's synthesized by the lipid kinase VPS34, and it was already known at the time, in 2013, that conditional neuronal-specific knockout models of VPS34 induced severe neurodegeneration early in the mice life, around 3 to 4 months. So the big question of my PhD project was to understand whether PI3P deficiency, as seen in AD patients, could drive endosomal anomalies and neurodegeneration in Alzheimer's disease. To tackle this question, we made use of a recently developed pharmacological inhibitor of VPS34, and we looked at the morphology of our early endosomes in neurons in vitro. And what we first detected was that these early endosomes were enlarged, as labeled by the marker early endosomal antigen 1, as was previously described for fibroblasts derived from AD patients. We later looked at late endocytic function, and in this case, we looked at organelles labeled by LAMP1, lysosomal-associated membrane protein 1, and we also looked at the lysosomal enzyme cathepsin D. While we didn't detect any major defect in the sorting of this enzyme to these late endocytic compartments, we noticed there was a significant reduction in the maturation of this enzyme, which typically requires the acidic pH of lysosomes. So this suggested that lysosomal function was impaired by PI3P depletion. And to confirm that, we also found that polyubiquitinated proteins and the autophagy receptor P62 accumulate when we inhibit VPS34 function, suggesting that proteins were being targeted for degradation, but they were not being processed efficiently. Going back to Alzheimer's disease specifically, we then focused on the amyloid precursor protein and its metabolites, and we found, while we found no changes in the levels of full-length APP, we found a particular increase in the APP C-terminal fragments. These are the fragments that are then processed into amyloid beta. And even though these fragments were accumulating, we didn't see an increase in the production of A beta, but rather a reduction. Since proteins are not the only substrates of lysosomal cleavage, we also looked at the lipid composition of neurons treated with a VPS34 inhibitor, and just like in AD patients, we saw accumulation of particular sphingolipids, namely ceramides, and also sphingomyelin. And very interestingly, we also looked at BMP, which is a lipid particularly enriched in late endocytic compartments. And even though the class itself was not significantly changed, we saw the accumulation of several species. And here you can confirm that BMP, stained in magenta or pink, is almost exclusively found inside the lumen of these late endocytic compartments. So we know from the previous literature that the amyloid precursor protein is sorted inside, it's internalized and sorted inside endosomes. And then as the endosome matures, part of it is secreted under the form of exosomes, and a significant part is also degraded in lysosomes. But since blocking VPS34 significantly impairs lysosomal function, could it be that it is now rather secreted under the form of exosomes? And that was indeed the case. We looked at the extracellular vesicle secreted by neurons in vitro after inhibition of VPS34, and not only we found an increase in markers typically associated with exosomes, namely ALEX and fultillins, but we also found a significant increase not only in full-length APP, but also APP-CTFs, polyubiquitinated proteins, and then again, P62. And I've got to tell you as well that the ratio of increase in exosomes was higher than the ratio of intracellular accumulation, really suggesting that these undigested lysosomal cargo was being targeted for secretion. And could these vesicles also contain a specific lipid composition? In order to address that, we repeated the lipidomic profile, this time of the vesicles alone, and we confirmed that these vesicles were accumulating, again, sphingolipids, in this case, the hydrosphingomyelin and glycosphingolipids, which, again, are also enriched in late endocytic compartments. And the most striking change was in BMP, the late endocytic phospholipid that I just mentioned. Finally, we confirmed these results using the in vivo neuronal specific knockout model. We collected these samples at 2 months of age, so at a pre-degenerative state, and we confirmed the accumulation of polyubiquitinated proteins and P62, suggesting again lysosomal dysfunction in these mice. We then looked at APP metabolism, and then again, we saw accumulation of APP C-terminal fragments, but not of full length or amyloid beta. When we looked at the lipid composition of the hippocampi from these mice, we saw, again, accumulation of sphingolipids, namely ceramides, and BMP again. Finally, these mice also showed increase in the secretion of exosome-associated APP C-terminal fragments and polyubiquitinated proteins. Altogether, we suggest a model in which VPS34 function and PI3P production is essential for regulating endosomal and lysosomal trafficking in neurons. When we decrease the levels of PI3P, we significantly impair the system, causing accumulation of a series of proteins and lipids, particularly APP, CTFs, and sphingolipids. And these undigested lysosomal cargo are then shuttled for secretion as a system for alleviating intracellular accumulation. And these exosomes, since they are enriched in APP, CTFs, and particular lipids, namely BNP, could potentially be used as markers of endolysosomal dysfunction, particularly in a stage before neurodegeneration. So our results lend support to the growing theory that lysosomal storage disorder is also a pathological hallmark of Alzheimer's disease or AD-like pathology on top of amyloid beta plaques and tau tangles, and suggests lysosome function to be a candidate therapeutic target for the management of Alzheimer's disease. Finally, I'd like to thank everyone who made this project possible, not only the people who worked, with whom I worked closely, but also everyone who created the program and funded the studies. Thank you all.